Good afternoon, everyone. Welcome to the Phi Sigma webinar. My name is Fan-Bing Kong, and I am the chair of the program committee. We have the topic is precision fermentation, principles, applications, and pathways to commercialization. And the speakers, we have Dr. Joseph Usack and Ms. Shalima Sreenath. And next month, in April 22, we have a webinar. Topic is disaster microbiology by Dr. Smith. And in May 5th, we have another webinar, which is insight from a new faculty, what I wish I knew before starting a faculty role. So just keep in mind our schedule. We will also post the webinar information on the publish on the newsletter and also post on the website. We'll also send a reminder to you before each webinar. Okay. So a brief introduction of today's speaker, Dr. Joseph Usack. Currently, Dr. Usack is an assistant professor at the UGA Department of Food Science and Technology, and he joined the department in 2023. And Dr. Usack's research involves bioprocessing engineering research with open cultural microbiome and gas fermentation to facilitate the recovery of resources within the food and agricultural sectors. And also his research involves life cycle assessments and techno-economic analysis. And Dr. Usack earned his master and Ph.D. from Cornell University Department of Biological and Environmental Engineering. Before arriving at UGA, Dr. Usack spent several years in Germany as a research group leader at the Sustainable Environmental Biotechnology Lab at the University of Tübingen, and as a guest researcher at the Max Planck Institute. So our second speaker, Ms. Shalima Sreenath, she holds a Master of Engineering from Cornell University and has over 10 years of experiences in the food biotechnology industry. And she currently, she's the head of Downstream Processing at Cellibre in San Diego, where she focuses on isolation and purification of novel small molecule isolation derived from precision fermentation. Prior to joining Cellibre, she has held process and project engineering roles at Kerry and Beyond Meat. So today, Dr. Usack will present the first, and then Mr. Sreenath will present. So we'll have a joint Q&A session after the two presentations, and so you can also type your questions in the chat box. So with that, we can start today's presentation. Dr. Usack, the floor is yours.
Thank you very much, Dr. Kong, and thank you very much, Phi Sigma, for this invitation to present. It's a great honor, really. So I hope you enjoy my presentation. Let me see if I can pull it up here. Okay. Okay. Is everyone able to see?
Yes.
The presentation PowerPoint? Okay, great. Well, I'll jump right in because I know we don't have a lot of time. I know that I think I promised to do a deep dive in precision fermentation, but I don't know if that's possible in 25 minutes. It's more like just putting your toes in the water. Yeah, my presentation is going to be about kind of defining what precision fermentation is, and also talk about some of the key principles and applications. And so just to give a little background about my research motivations and interests. So there was this report that was put out by the White House several years ago in the prior administration about where it sees the U.S. economy going and what technologies they will employ. And one of the major areas is biotechnology and biomanufacturing. They see, as well as many other scientists see, the great opportunity that lays there in this technology, not only for creating new bio-based products, but also for supporting new jobs, also helping to combat, environmental degradation, climate change, and that kind of thing. Around the same time, the Department of Energy also put out its own report where they did a census, essentially, of all the organic waste that's generated within the country. And they found that there's billions of tons of this material, and a lot of that material is currently underutilized. And this material can come from agricultural production and residues, food wastes, forestry waste, and that kind of thing. So it's a tremendous amount of material, and it's actually a resource, whereas historically it's often been considered a waste. It has carbon, it has nutrients, and it has energy in it. And so considering that, and then also considering the versatility of what we can do with biology, we see that there's a huge opportunity in this research space. And so this slide is showing essentially all that organic material here on the left side. And I just want to quickly ask, can you see my cursor, my mouse? Yeah, okay. So over here, we see this is all that billion tons of material. And again, it can be food wastes. It can be wastewaters from food processing. It can even be dead carbon in the form of CO2. And this comes as industrial smokestack CO2 or CO2 from fermentation processes and that kind of thing. But the idea is that we can use that material, feed them to a whole host of different types of microbes, and then use them to make all kinds of different products. And so this is just showing some examples of microbial protein-based bioproducts, everything from food ingredients to animal feeds, bioplastics, etc. And so there's some substrates that you can use directly, feeding them directly to the bacteria or other microbes. But there are very often more complex substrates, heterogeneous wastes that need to go through some pre-treatment steps in order to put them into a form that's more usable by the microbe. So breaking down complex molecules into more simple intermediates, and that's what this blue box is showing here. It's showing different physical and chemical conversion technologies, also biological conversion technologies. But you can chart many different paths from this left side of the chart to the right side of the chart, right? And that's the amazing aspect of this technology. And this little brief introduction to what I do in my lab. So I do wet lab work. So I develop bioprocesses in the lab with typically open culture microbiomes. I also work in gas fermentations. And also I develop experimental platforms that allow us to answer basic questions about these technologies. And then beyond that, besides just developing these systems, we are trying to scale up and further develop these bio-based products. So that's bio-based foods, bio-based products, and bio-based chemicals. And then finally, we also look at these technologies from a life cycle perspective. So we want to understand its sustainability in terms of environmental impacts and also its profitability. So we do techno-economic analysis around these technologies as well. Currently, I'm working on an experimental system for doing gas fermentation research. We want to use thermophilic acetogenic biocatalyst to convert CO2 using renewable power into food. We're also looking at ways of better integrating physical chemical pretreatments with some of these biological platforms. Some of the physical chemical treatments are pretty harsh and they lead to the production of toxic compounds, which pose problems during biological uses. So we're working in that area as well. And then another research area is to convert different types of food waste, including waste from, for example, the dairy industry, like acid whey, into bio-based chemicals. So we're focusing on producing these medium chain fatty acids as one target product. And then finally, we're trying to upscale a technology that I've been working on in Germany. It's a two-stage CO2 to protein process. And we are looking at specific applications for addressing food insecurity in countries like Africa. So anyway, that was a little bit about my research background and interest. So now I'm going to talk a little about fermentation. It's important to define fermentation before we try to define precision fermentation. So I think, as all of you are aware, fermentation has been around for a very long time, has a long history, and in the past it had been used primarily for food transformation and preservation. And there are many examples. This is a picture of winemaking from ancient Egypt in 18th dynasty. Sorry. Here's a picture of a baker making bread in medieval Europe, and then there's also evidence of cheese making as far back as 2800 to 2600 BCE. This is in ancient Sumerian civilization. So yeah, it has a long history. In the technology, fermentation technology has been progressing steadily through time, finding new applications, and also we're developing a greater understanding of how to fully harness it. And a key turning point was in the middle 19th century when Louis Pasteur just kind of discovered that microbes are responsible for this process, and so that led to a lot of new research. We no longer looked at the fermentation process as like a black box. Now we start to actually use it as a tool that we can manipulate. And so over the years, in the last several decades, fermentation has seen many new applications beyond just food modification and preservation. So it's now being used for producing things like biofuels and biochemicals. It's being used by the pharmaceutical industry for making antibiotics, antibodies, enzymes. It's also being used to produce specific food ingredients and supplements and vitamins. So the array of products is growing year by year. We're also looking at new ways of supplying fermentation with different feedstocks. So historically, the feedstocks have been what we call first generation feedstocks, which means they're using things like sugar, which compete for food production. But new technology areas are now looking into second, third generation feedstocks, such as gas streams that contain CO2, or organic wastes, and then also looking at different products that can be developed from those feedstocks. So I want to start by defining fermentation in its traditional sense. So fermentation is a metabolic process in which microorganisms such as bacteria, yeast, convert organic compounds into simpler compounds in the absence of oxygen. So these are anaerobic environments, and it's used by the microbes to generate ATP through substrate level phosphorylation. So I wanted to define that fermentation that way because the current use of the term has kind of moved away from that traditional definition, and it means a completely different thing now. But yeah, that's important to point out that it traditionally is about a certain type of metabolism that takes place in anaerobic conditions. A more simple definition was put forward by Louis Pasteur. He, excuse my French here, but he says la fermentation is la vie sans l'air. So that translates as fermentation is life without air. So yeah, and this is, I think you guys probably remember from microbiology or biochemistry that there are these two major pathways for producing energy at the cellular level. Either you're doing it through aerobic respiration or you're doing it through a fermentation process. So again, here we're focusing on fermentation. Okay, so in fermentation, so I should point out that now we are not, when we use the term fermentation, it doesn't just mean anaerobic metabolism, it's also incorporating aerobic respiration, and I'll give some examples of that later. But in this field, we are able to categorize fermentation into three basic groups. So I already mentioned traditional fermentation. This is where microbes are modifying the food itself. So again, taking milk, turning it into something like cheese, or taking like cabbage or vegetables and turning it into sauerkraut, right? Another type of fermentation is what's called biomass fermentation. And here, microbes serve as the food themselves. So we're actually harvesting microbial cells and then consuming those cells or major cell components like their proteins or lipids. And then precision fermentation. So precision fermentation is different in that the microbes are actually being used as factories to produce specific metabolites or biocompounds, and these are serving as food ingredients. So these are three general categories of fermentation that we're kind of now using in common parlance. And then there's other ways of doing fermentation. There's different approaches. You can do what's called solid state fermentation. In here, microbes are actually grown on a substrate, a solid substrate like agricultural residue, like spent brewer's grain or wood chips or that kind of thing, and they use that as their feed stock. And then there's what we call submerged fermentation. Here, microbes are growing in a liquid, typically in a solubilized liquid substrate. And then there's also gas fermentation. So here, this typically takes place also in a liquid bioreactor, but the bacteria are actually feeding on gases. That's their substrate. So you might be aware that there are microbes growing at the bottom of the ocean around these subthermal vents, right, that are spewing out hydrogen gas. So microbes can actually use gases as a substrate. Okay, yeah, and so there are different what we call microbial hosts for doing these fermentations. We like to use the term biocatalysts. We borrowed the term catalyst from the chemical engineering industry, but here, yeah, we're really using the microbe to carry out certain bioprocesses or reactions. So, and there are four general groups, and they're typically single-cell organisms. We can use bacteria like E. coli, for example. Yeast is very commonly used as a host. Filamentous fungi, and then also algae. So these are what would become our cell factories for producing our fermentation products. Okay, so this give a quick workflow of what solid state fermentation looks like. So again, using a solid substrate as the growth media. And here are some common examples. You have corn stalk, soy residue, also spent brewer's grain is another common one. And you use that to create this bed that you can inoculate, typically with a fungus. And so that you see here, somebody's spreading this substrate on a tray and then putting these trays in this large growth chamber where the temperature and the humidity is controlled. And they're growing basically a bunch of mycelium or fungal biomass that can then be harvested. You can see very clearly in this picture over here that fungal growth that grows on top of the solid substrate. So it's quite a lot of material. It can be harvested, cut up, and then further processed into, for example, alternative protein-based foods. So this is a company, Atlas. It's making a bacon alternative using this type of fungus. So submerged biomass fermentation, this is probably what you're more familiar with. This is how you make your beer, how you make wine. So, typically you start out with some simple sugar substrate that's fed to a bioreactor that contains your liquid where the bacteria grow. And like I said, these are what we call first generation feedstocks, because again, they're competing with food production. But you grow a bunch of that of the biomass in the bioreactor, either to use as bacterial cells or their products, like for example ethanol or lactic acid. But then that material has to be harvested. And so this is a what we call a U-loop airlift fermentation system. It's used by the company Corn to make this filamentous fungi biomass. You can see here this picture of them harvesting that biomass from the reactor process. This is something that's desired for its high protein content, and also the filamentous fungi grows in such a way that it can, after some processing, be turned into meat alternatives because of its meat-like texture. And so there are several companies that are using this approach to produce alternative meat products. Like I said, corn is a very common, or has been around for many decades, has had a lot of success, but more and more companies are coming online. Okay, and then finally, gas fermentation. So, like I said, you can use gases as a substrate to grow microbes, and this is where we get into the area of using second generation feedstock. So we can use CO2 and hydrogen, for example, to grow acetogenic bacteria, and also archaea. You can also use microbes that consume methane or natural gas, like these methanotrophs. And there's also bacteria that can grow on hydrogen, oxygen, and CO2. And again, you can either use them to produce microbial biomass, which you then harvest for feed ingredients or even human foods, or you can use them in the context of precision fermentation. Although there's not very many examples of that, but there are a few. So here is one company, it's based in Finland, it's called Solar Foods. They are using a type of null gas bacteria, which feeds on hydrogen, oxygen, and CO2. And they're using that to produce a protein powder. So they're extracting the protein from the microbial biomass. Another company that's actually been around for also quite a few decades is Unique Protein. They're also making a protein powder, but their main application for the powder is an animal feed. But these are, some commercial examples of that technology. Okay, so that brings me to precision fermentation. I should point out that this term, precision fermentation, is not indicative of a new technology. It's actually a rebranding of a technology that's been around for several decades. It was previously known as synthetic biology, and but I think as the industry started to look at using the technology to make food products, they want to get away from this term synthetic, right? And so they came up with precision fermentation because it's I don't know, a little bit more palatable, a little bit more familiar. But the technology is not new; it's been around for decades. And I like this particular definition. It's the insertion of target genes into a microbial host to produce specific biomolecules through controlled fermentation processes. And this is allowing for the production or biosynthesis of compounds that you typically find in other organisms, like plants, animals, for example. The technology, like I said, is not new. It has been used and approved decades ago. One common example of the technology is the production of rennet, which was approved by the FDA in 1990. So rather than harvesting rennet from animals, they are now producing it through the fermentation process. It's less costly. You end up with a more consistent product, and it's also more humane. So that's an example. There's also another term called cellular agriculture. So precision fermentation is a form of cellular agriculture, but cellular agriculture also includes cell culturing, and this is different. Cell culturing is where you're actually using animal or plant cells directly to produce more of those same cells. So it's like cultured meat, for example. And here's an example of a company that's using chicken cell line to produce basically chicken alternatives. Okay, and then very, very finally, I want to say that precision fermentation is not the same as molecular farming. Molecular farming is where you're taking a host like, I don't know, a plant, and then modifying it to produce certain biocompounds. Some examples of those are, this is a company called Moolec that's has genetically modified the soy plant to produce porcine proteins, and then they're harvesting those express proteins from the soy plant to produce food ingredients. Another example is Nobel Foods. They have also modified the soybean plant to produce casein protein, and that casein protein they extract and then are using to produce different cheeses. So how does precision fermentation work? This is a very basic workflow. So essentially, you choose a microbe or a host organism. You select a gene and you incorporate that gene into your host. You add both of those components to a bioreactor along with some feedstock. Like you said, it's typically sugar, and then you grow a bunch of that microbe, and that microbe will express your target compound, which you later extract through downstream processing. And this is a picture of a fermenter. This is what you might see at a company that's doing precision fermentation. But yeah, so it's very key to point out that with precision fermentation, you are trying to produce specific functional biochemicals, and that the technology requires, sterile conditions, also uniform feedstocks and growth media. These are all major cost drivers in the technology, but I won't get into that at this point. And then also the process is run in batch, so it's not run continuously. And this is done to avoid contamination or genetic drift in your host organism. Yeah, and then downstream processing is required to extract your product for further use. Okay, so this is looking a little bit closer at the upstream processing. So and using an example of producing recombinant protein in precision fermentation. So for example, what you do is you choose some DNA donor. Say, for example, you want to produce a whey protein. So you find the gene from your DNA donor that codes for the production of casein. You then choose a vector. It could be like a plasmid. You open the vector with a restrictant enzyme, and then you incorporate the gene into that molecule, and then that molecule is introduced to your host cell, and then you grow that host cell in one of these fermentation bioreactors, right? And you produce a bunch of biomass, and that in those host cells will produce your target product. They'll express that product. And in this case, it's a recombinant protein. And so once you have that whole process worked out, you basically use that to inoculate a large bioreactor, and run the bioreactor with these other components, your feedstock, your nutrient media, and also you control the reactor temperature in mixing and that kind of thing, and produce a bunch of that product. And then you go through a whole series of downstream processes, which I'm not going to get into, but maybe Shalima will cover in her section. But you have to basically extract that product from either the fermentation broth, if it's an extracellular product, or from the microbial cells, if it's an intracellular product. But then you have your target ingredient at the end of the day that you can use for formulating different types of food products. And so I'm not going to go through this particular slide in detail, but I just want to show you that there are, this is what's called biomass fermentation. So here you're, again, just producing microbial cells that you harvest as a bulk material. And then precision fermentation, you're typically producing metabolites or specific biocompounds. And again, they can be proteins, they can be lipids, they can also be other things entirely. They can be certain vitamins, for example. And then one key consideration is whether that product is excreted from the cell or if it's contained within the cell. It has implications for how that material is processed downstream of the fermentation. And so I just want to point out a few companies that you might be familiar with that are kind of leading the industrial landscape. So Every is one company, used to be Clara Foods, I believe. They are producing egg white proteins. There's also Perfect Day. They are producing milk proteins like casein and whey to make dairy alternatives like ice creams and yogurts and that kind of thing. And then there's also Geltor. They're making collagen proteins. And finally, there's companies like Impossible and Triton that are producing heme proteins for producing things like the Impossible Burger. Yeah, and so that's just those are just a subset of all the companies that are out there. This is a figure from a recent opinion article showing different host strains that have been used, and then also the companies that are employing them, and also the products that they're generating, and finally the some of the properties that they're trying to confer into their food items. So as you can see, it's quite a big area. There's a lot of different products you can produce. using this technology. And yeah, and this is the whole alternative protein landscape is just again, protein is just one biocompound that you can produce through precision fermentation, and so it's a very promising field. There's a lot of interest. It's getting a lot of support from different agencies, both venture capitalists, also from big food companies, and also from governmental bodies. So I think it's a promising future for this technology as a whole. So I think that's all I had. Hope I didn't take too much time, but thank you very much for your attention.
So yeah, thank you, Dr. Usack, for that overview about fermentation and precision fermentation, and explaining the basics of precision fermentation. So what I would like to cover in my talk would be, once you have a strain which is engineered and it's making gram per liter quantities of your target ingredient, how do you scale that up from like the lab to commercial, and what challenges do you encounter there, and what all do you need to keep in mind when we are developing a process from like lab to pilot to commercial? So I like to think about precision fermentation as having like three pillars. So as Dr. Usack has mentioned, so the first one is strain engineering. So that involves the engineering the microbes to make them produce the target molecule. So that is the whole recombinant DNA technology, where you are engineering the strain and making it ready to make the target molecule. The second one is fermentation. So once you have the strain ready and engineered, you grow and multiply them, basically like any other fermentation. So you grow them and multiply them, and also you keep account of like how much of the target product the engineered microbes are making. So this can go from 250 mils of shake flask all the way up to like one liter of on the bench, like 1 liter. And then like as we go into commercial, it can go higher in volumes. And once you are making the product in the tank, now you have the product in the tank with the cells, the media components, any other impurities the cells would have excreted. So the goal of downstream processing then is to purify and isolate the target molecule from the cells and all the other stuff which is in the tank. So that's the third pillar, as I would call, which is downstream processing. So for process development, I consider process development and precision fermentation as a continuous feedback loop. So each of these streams would have to like get the feedback from how it is going. So strain engineering would need to take the feedback on how fermentation and downstream processing is going. And then from fermentation, like you would need to understand like how the molecules are, or the cells are performing, and how well it is generating the molecule. And then for downstream processing, you would need to know like how well you're able to separate and isolate these molecules, and then provide feedback back to the strain engineering and fermentation groups. So as far as process development goes, in strain engineering, host selection, that's like one of the main key components when selecting the right microbe for the target product. So you have a product you want to make, and you need to select like, is it a bacteria, a yeast, a fungi? Like what is going to be the best suited host, which can make the target molecule with like minimal byproducts, and then at the same time, like we need to make sure like it doesn't kill itself in the process. Another thing is strain engineers spend a lot of time optimizing gene expression and transport of the product. So if they identify a gene, and if they knock it out, and then they see that the product production has increased by the cell, they try to put in like more copies of the gene or take away more to see like how that is going to affect the final production of the final product. Another one is transport. So as our target product is made in the cell, we need to make sure that it is transported out of the cell and secreted ideally, and at the same time, it doesn't like accumulate inside the cell and prove toxic for the actual cell itself. So that's like another area of process development which strain engineers spend a lot of time in. The next one is strain stability. So strain stability means that once you have an engineered strain, you need to make sure that even if you grow the cell for multiple generations, the cell is still going to maintain the engineered traits, and it doesn't like mutate in that process. So those are the three main strain engineering process development items, but most of the strain engineering is done on the bench, and then you move to fermentation. So for fermentation, the main goal would be titer. So titer is what you call as gram per liters of the product which is made. So how many grams of your target product is being made in like 1 liter of fermentation broth. So this is what we typically pay attention to know if a fermentation is like going successfully or not. So higher the titer, the better. The next one is like optimizing, like any other fermentation. For precision fermentation as well, we need to like optimize the pH. So optimization of pH, one thing is to make sure the cells grow well, but also like if there are other byproducts which are made in the by the cell, sometimes tweaking the pH can get rid of like some of the the derailment products which is made by the cell pathway. And then temperature is one. So we need to find out like what the ideal optimum temperature is. Aeration is another. So if your microbe is aerobic, you need to make sure it has enough oxygen supply to keep growing as well as make the target molecule. And also feeding strategies like do you supply the feed continuously, or do you put in a bolus in the beginning and then call it good? So all of these are things that fermentation engineers would work on to optimize the process. Another one is cell density. So sometimes you can see that the cells grow and get really fat, and now you have an issue with that interfering with the product, and that typically it becomes an issue in the downstream processing. So you need to make sure that the cell size doesn't impede the ability to isolate the products in downstream processing. And then the other one, like I mentioned, is host cell toxicity. So we need to make sure that as the cell accumulates the product, it doesn't reach a point where you're trying to make this product A, but is there a level of product A above which the cells will die? So that's something like we need to understand as well. As far as downstream processing go, one of the main thing is like if the molecule which is produced, if it's intracellular or if it's secreted out of the cell. So that will determine if we need like a lysis step in the beginning. So do we need to break open the cells to release our target molecule out, or can we get rid of the cells and then just deal with the remaining media components and the product? Another one is, does the molecule separate based on density? So if it's, if it's heavier or lighter than the cells, centrifugation is a good option to remove the cells. So you could either like, centrifuge and remove the cells and deal with the broth, or centrifuge the product and the cells and then get rid of the media components that way. Another one is if the particle size of our molecule is different than the cells or like other impurities, you can use filtration there. Another downstream operation is liquid-liquid extraction. So this is if your target ingredient extracts into a solvent. As with extraction of any other component from like a plant, the same applies to a cell, to a molecule which is made by a cell as well. So can you use a solvent and extract it from the biomass using liquid-liquid extraction or liquid-solid extraction? And then the other one is, can you use ionic charge, hydrophobicity, or specific binding characteristics? So this would be the use of chromatography. So in most of the cases, if you need like really high purities, chromatography is typically involved. But of course, it comes with a cost. I briefly want to touch base about downstream processing. So this is like a picture from my lab. So we are working on like isolating a molecule. So this is what the fermentation broth looks like at the end of the fermentation. So this has the product in like gram per liter quantities, but then as you can see, it has a lot of cells, a lot of media components, and it's like it looks way different than our final product, which is this. So you subject this broth through a series of downstream unit operations and then end up with this product, which is like highly pure. Usually downstream processing is one of the last thought of steps in process development, because most of the focus is on making the product in the fermentation tank itself. But we need to keep in mind that the end format of the product dictates its usability in the target application. So unless you have the product in the desired form, you cannot use it in the target applications. And also, it could be that downstream processing can significantly contribute to the final costs of the product. So a majority of processing costs, like the main one is fermentation, but then downstream processing also adds a lot of cost when you're looking at scaling up. And then once the target purity is achieved, then it is a balance between like yield and purity. So if you want like say like greater than 98% pure product, you need to make sure that you are not purifying it away so that you end up with only say 30% of your starting material. So it's a balance between like how pure you want your product to be and then how much yield do you get when you're processing it through the downstream process. Then scaling up. So once you have developed a process in the lab, like I said, so you have the strain engineering done, the fermentation done in like a 1 liter fermenter, and then you have a basic downstream process developed in the lab. What all do you look for when you go to like a bigger scale? So a general rule of thumb is that you do a 10x increase in scale. So you go from like a 1 liter reactor to a 10 liter to 100 liter, and then so on and so forth. Most of the times, like this is the ideal case, but most of the times, depending on how much resources you have, like what your timeline is, you may skip some of these stages, and also depends on like how representative is the data you get from 1 liter and how well you think it's going to translate to a bigger reactor. But that's typically like the scale you would look at. So 10 liter and 100 liter, they are mainly pilot. Even 1,000 is sometimes considered pilot scale. And then anything larger than that is considered commercial scale. And it can go higher than 10,000, like 100,000 as well. So for fermentation, one main thing is like temperature and pH control. So as these microbes are growing and releasing heat, we want to make sure the temperature is maintained. So we need to make sure all the heating, chilling systems have the capacity to cool the fermenter down and then maintain the temperature. The same with pH. Imagine a really large tank with a pH probe on one end. We need to make sure the components are like mixed well, so that the pH probe gets a really good reading. And if you're adding like an acid or a base to control the pH, we need to make sure to give it like enough time and enough mixing so that the pH, whatever the pH probe is reading, is representative. Sometimes you may have like multiple pH probes at like different locations in the tank, so that you remove that variability. The other one, which is the main one, is aeration. So depending on like the oxygen uptake rate of your organism, so the organism would need like a certain amount of oxygen to survive and thrive. We want to make sure that we are producing more oxygen than it requires, or like at least enough oxygen so that it doesn't die. So that is called the oxygen transfer rate. So we want to make sure like by providing like air, or in some cases, we even provide like oxygen, that there is enough oxygen available in the tank at a certain moment for the cells to use up. Agitation is another one. So as we are, providing the oxygen by like sparging, the agitation makes sure that the oxygen bubbles are like broken down and dispersed. Agitation also makes sure that the contents of this tank is like well mixed, but at the same time you need to make sure that the mixing is not too vigorous. Otherwise, like if the cells have shear sensitivities, that might be an issue, and the cells might be compromised. And at the same time, with aeration and agitation, one main issue I have seen is with foaming. So it's not like we can provide as much air and agitation as we want. At that point, like foaming becomes an issue, and you don't want so much foaming in the tank. Another one, like I mentioned, for foaming, what we can also do is like add anti-foam, but at the same time, we want to make sure that we keep a track of like what all other processing aids we add in the fermenter, because eventually we would need to remove it through further processing downstream. So it kind of adds cost. If you're taking care of like one problem upstream, then it could end up as a problem like downstream. Another one is raw material sourcing and quality. So most of the times when we are doing proof of concept tests in the lab, like we use high quality materials, like even sugar sources, sometimes like high quality dextrose is used. But then as you go up in scale, that becomes expensive. So we need to think about like other what is realistic for like bulk purchase. Like, is it corn syrup, or is it like a waste feedstock, which would be economical to as we move up in scale. For downstream processing, as we go up in scale, the major point which comes in is like batch mode versus continuous. So as you are developing a process in the lab, you do it batch mode. So think of like if you're centrifuging a culture, like you will take it in a bottle and then you'll spin it in the lab, and then you get like a pellet and the supernatant, which is like clearly separated from each other. But then in the at scale, like if you're using a continuous centrifuge, we need to make sure that the pellet is pumpable, that a pump can like take it from one place to another. So in that case, the pellet will have like more water come through, which could mean that if there is like a product in the liquid phase, like more of the product is lost to the pellet. So that is one thing which we need to keep in mind when it's like batch versus continuous. The same goes with filtration. It is easy to filter like a liter and collect the product, but then like at scale, if you have to maintain the filtration going on, like you need to be aware of the flux. So how much material passes through the filter before the filter fouls. So that's where like balancing throughput with quality comes in, because you want to make sure that you can increase the flux by using a larger pore-sized membrane, for example. But then do you end up like passing other materials through which you don't want to? So it is a constant battle of balancing throughput at scale with quality. The next one is product stability. So if you're exposing your molecule to like high temperatures and pH conditions or any extreme conditions, how stable is your target molecule going to be as it goes through downstream processing? The next one is reactor design. So this one is not referring to the fermenter itself. It's mainly referring to the tanks which we use in downstream processing. So do the tanks have like really high shear agitators, or do they have efficient mixing? If you're doing acid-base reactions, like does it maintain efficient mixing, and at the same time, like does it create more foam? Again, like particle size in reactors. So this is especially if you're doing a precipitation reaction. We need to make sure that if it's a big tank and you're adding your like precipitant like in a slow rate and it's getting mixed, how does the particle size grow? Like does it nucleate or is it does it grow by particle size growth? Another one is flammability. So when you're developing a process using solvents, especially in the lab, we can use like solvents which are very effective in separating molecules. So a solvent like ethanol or ethyl acetate, these are commonly used solvents. But as you scale up and move to a lab, move to a plant, we need to make sure that the whole building or like the entire processing area is rated for explosion proof. It's explosion proof, so that there is no risk of explosion when the solvent vapors are present. Another one is solvents, since they add cost, we need to make sure wherever possible, the solvents can be recycled, as well as like any other processing aids can be recycled. And then again, minimizing waste. So a lot of wastewater is generated as you go through downstream processing. So we need to be conscious of like how much waste we end up with as we go through this. Main cost drivers and challenges. So from like whatever I mentioned here, the main cost drivers in strain engineering, one is like extensive R&D resources. The main cost comes from just time taken for like engineering a strain, testing it out, and then reiterating the process. So from strain engineering, it's mostly the time taken for like engineering the strains. For fermentation, it's input optimization. Like I mentioned, we need to make sure like all the media components is like it's cost effective as we move up in scale. Toxicity is more of a challenge. It's more than a cost driver. It's like a challenge that if the target molecule ends up being toxic, then you have to go back to strain engineering and then start the whole process again. With downstream processing, the main cost drivers are equipment. So if you need like any specialized pieces of equipment, say like a specialized piece of filter that you need, like all of that can add cost to your process. And especially if like a filter needs to be replaced every now and then that needs to, that'll add cost. And then like I mentioned, product stability is one thing. So that's where techno-economic analysis comes into play. So what techno-economic analysis does is it looks at the whole picture holistically to know what all are the factors which input costs into your process, and how can we understand like what factors like provide the major costs, and then go from there as you go towards commercialization. So this graphic, it shows like the main cost drivers as you're looking at precision fermentation. So you can see that it starts with CapEx and OpEx. So that would mean like capital expenditure. So capital expenditure is like any equipment or building costs, like anything which you're, which is brought new, or like a process line expansion, any new piece of equipment which is installed, all of that goes into a capital expenditure. And then OpEx would be like the operational expenditure, like how much like utilities do you use, how much do you spend in labor. So all of that is counted in OpEx. So between like CapEx and OpEx, another thing the techno-economic analysis can tell us is that what scale do you need to go to so that your product becomes cost effective. So is sometimes like you may just be, you may be good even at like 1,000-liter scale, but as the scale increases, the cost, the CapEx and the OpEx cost comes down. So sometimes you may need to go to 100,000-liter scale to be cost effective. So that's what the CapEx and OpEx from a TEA would tell us. And then also like, would it make sense to put automation in, or would it be fine without? That's like another thing. As far as fermentation goes, what is the target grams per liter? So once you have the flexibility to change the gram per liter in your TEA, you'll be able to understand what is like, is it 10 grams per liter? Is it like 1 gram per liter? Or do you need like 100 grams per liter of your product for your process to be cost effective? Then of course, like productivity comes into play. So if you're targeting, say, 10 grams per liter, and if your model says that you'll reach that 10 grams per liter only in like 10 days, whereas in like 5 days you reach like 8 grams per liter, that is like a significant, that's a hit you can take, because it'll significantly drive down the cost because you're spending less time in the tank in the production facility. And like I said, the cost of goods sold. So you'll need to optimize the feedstock selection, and then also consider circular economy strategies like making use of waste stream. That's where it becomes really attractive. If you're able to make use of the carbon sources from industrial byproducts, that would really bring down your cost of goods. Plus, it's also good from like an environmental and sustainability standpoint. And then DSP, the yield and purity. So how pure do you need your product to be? Do you need to be like 99.9% pure, or is 90% okay, provided that you know what the other 10% is? And then so identifying if you can cut down any of the downstream processes based on the purity requirements. And then finally, the market viability. So comparing the product which is derived through precision fermentation to what is out there in the market right now, something which is derived from plants or animals, what is the cost of that product, and how long do you think your precision fermentation product will take to reach cost parity? That's something like we'll need to look at while going commercial. And then regulatory approvals. So this comes in if there is like any specific testing which would have to be done every time, and also supply chain, like if there is if your raw materials are locally available, or if you need specialized raw materials which are imported, that all that adds up to the cost. So I would like to end with this graphic, which I really like. It shows the evolution of biomanufacturing and like a future lookout. So it shows the different categories of products which are biomanufactured. So at one hand, you have your pharma biologics, which are already at cost parity. So the green is the current production price, and then the orange is the non-bio alternative. Like so. And then the orange would be our target. And then you can see that for pharma, through precision fermentation, we are already at cost parity. And then for specialty chemicals, we are almost there. Food is right here. It is almost half there. So as we move towards 2040, the projection is that you achieve cost parity for food as well when compared to bio alternatives. And then the last would be like cheaper chemical precursors. That's also an avenue which is being looked into for biomanufacturing. So that's like a hopeful lookout. That's it. Thank you everyone for listening to my talk. Happy to take any questions.
Well, thank you very much, Dr. Usack and Ms. Sreenath. Very insightful, comprehensive presentation. We can see the questions in the chat box. There are a few questions. I think the first question, Anton's question, after the host has been selected, how are host cells modified to deliver the specific fermentation requirements? What is the gene expression? Maybe both of you, you can answer this question.
I can. You want to?
No, you go ahead. I can add in. Yeah.
Yeah, I mean, it's that's a whole lecture in itself. How that process works. But yeah, so the gene insertion or gene encoding process, like for example, for a desired protein like casein or insulin, it's inserted to the host cell using techniques like CRISPR, or also homologous recombination. Plasmid transformation is another approach. And then the second part of your question was, what is gene expression? Yeah, so again, so the idea is once the gene's already in your host organism, you want that gene to be turned on so that it starts producing that particular product. So if it's a protein, it's producing that protein that you can then later extract using downstream processes. Do you want to add anything to that, Shalima?
Yeah, just the modification process. So the way it typically works is that once you have the genome, you can get like guide RNAs, which will have the protein, the Cas9 protein, which acts like a scissors to cut the gene in the side place. So once you add that and electroporate the cells, the protein would basically like make the desired cuts in the specific genome sequence, and then like add in your desired gene, which you want it to express. So that's kind of how it works. Like, so you have the genome, and then it cuts, and then basically inserts like a sequence in, to put it in simple terms.
Thank you. Another question, Kostov's question, is strain engineering truly essential for precision fermentation? While it's traditionally the go-to approach, I'm curious about your thoughts on leveraging alternatives or complementary strategy for producing specific compounds. Could the quorum sensing offer more dynamic or efficient control in certain applications?
Mm-hmm. That's a tricky one. I mean, precision fermentation, it kind of depends on how strictly you define precision fermentation. And I mean, for me, definitionally, precision fermentation requires engineering of specific strains. That's, I think, where the part precision comes in. But I should mention that that's not the only way to produce specific biocompounds. So like I mentioned that I do open culture fermentations. And so there you're working with a microbiome. And you can manipulate the microbiome using environmental pressures like pH and temperature, nutrient limitations, in order to force that microbiome to produce very specific products. So we are producing medium chain fatty acids with very high specificities, and that's using thousands of bacteria in a community, right? So it's not a single organism, and I'm sure that they are using quorum sensing, as part of that whole ecosystem process. So yeah, precision fermentation is not the only way to go, I guess, is my short answer.
Yeah, I would like to add that my thought is that it's called precision fermentation when strain engineering is involved, but traditional fermentation, that's an option as well. Like you mentioned, modifying the environmental factors and making the microbe produce that. So that's an option as well. But when it's called precision fermentation, it typically involves strain engineering.
Okay. Cherry's question, will the products from precision fermentation be subjected to special regulation? Is it considered GMO product?
Yeah, so on that one, so it is typically, so right now it is not considered GMO product, provided that the engineered strain is not present in the final product. So the strain is engineered to make the final product, but then your final product, which the consumer is consuming, does not have the genetically modified organism in it. So it is a kind of a gray area because you are using genetic modification to make the product, but then the genetically modified organism itself is not present in the final product. So it is not considered GMO.
Okay. The last question, Miriam asked, will we receive the recordings of this webinar? I can answer yes. We're going to post the video on YouTube.